Dear colleagues, first of all, I would like to thank the possibility to take my presentation on this excellent conference about the following topic: cyclodextrin in traditional and alternative drug formulation. During my presentation, I will give an overview about the pharmaceutical application of cyclodextrin and introduce the results of our team at University of Szeged Institute of Pharmaceutical Technology and Regulatory Affairs. How connect exactly this topic to our research work? Let me introduce you the main research interest of me. Basically, we apply different kind of technological procedure to improve the solubility, dissolution rate, permeability, and bioavailability of poorly water-soluble drugs with the use of, for example, cyclodextrins and polymers. In the field of particle engineering, our aims develop the technological protocols, especially for alternative drug administrations such as nasal and pulmonary. The content of my presentation will be the following. I will give a basic overview about cyclodextrin, including their pharmaceutical application. I will talk about the cyclodextrin-based drug delivery and innovative delivery. Then I will present the results of our case studies where cyclodextrin were applied through the formulation development. The cyclodextrin comprised a family of 3 well-known produced major and several minor cyclic oxysaccharide. The 3 major cyclodextrins are crystalline, homogeneous, non-hygroscopic substances which are torus-like microrings built up from glucopyranose units. In an aqueous solution, the slightly apolar cyclodextrin cavity is occupied by water molecules which are thermodynamically unfavored and therefore can be readily replaced by appropriate hydrophobic gas molecules. The stoichiometry of drug cyclodextrin complexes is most frequently 1-to-1. However, natural cyclodextrins have a tendency to self-assemble and form aggregates in aqueous media. Cyclodextrin aggregation can limit their solubility. Through derivative formation, it is possible to enhance their solubility and complexation capacity, but these depend on the type of substituent and degree of substitution. Cyclodextrin and their derivatives have received considerable attention in the pharmaceutical field, and an increased number of reviews have been dedicated to their industrial and pharmaceutical applications, as you see here on this slide. Various physicochemical properties of drugs can be altered through cyclodextrin complexation, especially drug solubility in aqueous biological media. In aqueous media, drug molecules of appropriate size and structure will enter into the central cavity of cyclodextrin molecules to form water-soluble complexes, and the frequently enhanced total drug solubility is observed. The various applications of cyclodextrin as solubilizing agents are summarized in the table. Cyclodextrin enhances the bioavailability of insoluble drugs by increasing its drug solubility, dissolution, and drug permeability. The feasibility of pharmaceutical formulation can be limited by stability issues, especially for aqueous formation of drugs that are prone to hydrolysis or oxidation. The reaction rates can be affected by inclusion of the chemical liable moiety of the drug into the cyclodextrin cavity. The cyclodextrin will also reduce denaturation in the peptide and protein formulations. There has been a growing interest in developing the rate or time control type oral preparations because an appropriate drug release from the dosage forms is of critical importance in realizing their therapeutic efficacy. Presently, the commercial viability of cyclodextrin-based oral formulations has been established. Cyclodextrin are supposed to act only as carrier materials and thereby helps to transport the drug through an aqueous medium to the lipophilic absorption surface in the gastrointestinal tract. The cyclodextrin enhances the bioavailability of insoluble drugs. This is achieved by making the drug available at the surface of the biological barrier, for example, skin mucosa or the cornea, from where it partitions into the membrane without disrupting the lipid layer of the barrier. So formation of water-soluble drug CD complexes can increase drug permeation through the biological membranes. To maximize drug permeation, the amount of added cyclodextrin into pharmaceutical preparation has to be optimized. The presence of pharmaceutical excipients can influence the solubilizing abilities of cyclodextrins, but this depends on the excipient's physicochemical properties. The nasal route is another effective way to bypass fast metabolism. In order to enter the systemic circulation, the drug has to dissolve in the aqueous nasal fluids. In nasal formulation, cyclodextrins are normally used to increase the aqueous solubility of lipophilic drugs. The lipophilic cyclodextrin acts as penetration enhancers, especially in nasal delivery of peptides. Pulmonary drug delivery can be limited by low aqueous solubility and slow drug dissolution. Insoluble particles are removed from the lungs by the mucociliary clearance in the upper airways and by macrophages in the alveoli. Cyclodextrin can increase the solubility, stability, and dissolution rate of water-insoluble and chemically unstable drugs, thereby leading to decreased clearance, increased drug absorption, and faster onset of drug action. CDs have applications in the design of some novel delivery systems like liposomes, microspheres, osmotic pump, peptide and protein delivery, specific site targeting, and nanoparticles. CDs have become more commonly used functional materials in nanoparticle-based drug delivery. The properties of nanoparticles can be advantageously modified by the inclusion of cyclodextrin or their derivatives. CD-conjugated nanoparticles have many benefits, such as improved drug solubility, and serve as drug carriers to specific locations such as cancer cells, which reduce toxicity to normal cells. In addition, CDs can overcome the limitation of nanoparticles, such as low encapsulation efficacy and drug loading. And now let's see our case studies where we apply different kind of cyclodextrin in drug formulation. Nifluminic acid is an anthracic acid derivative which frequently used anti-inflammatory drug, which also has a weak analgesic effect, and is primarily used to treat different forms of rheumatism, rheumatoid arthritis, arthrosis, and to decrease other inflammatory phenomena. It has some side effects such as nausea or vomiting, and in view of the pure water solubility of pharmaceutical ingredients, our aim was to increase its solubility and dissolution rate by applying different formulation methods. Cyclodextrin binary and ternary complexes were prepared using niflamic acid and hydroxypropyl beta cyclodextrin, and in ternary complexes we use polyvinyl pyrrolidone. The products were prepared in 4 different mole ratios: 2:1, 1:1, 1:2, and 1:3. We prepared, of course, physical mixtures as a reference samples. During the solvent method, we applied a kneading and ultrasonications, and of course, after the evaporation of the solvent, which was ethanol, we get the dry products. During the ternary system formulations, we prepared first the physical mixtures, after we used the kneading procedures. Then let's see the investigation techniques. We investigated the characteristic physicochemical properties, biopharmaceutical behavior for solubility and dissolution rate, and structural characteristics of the samples on the basis of the following: preformulation studies, in vitro investigation, and structural evaluation. Let's start with the physicochemical characterizations, where we check the wettability and the N-octanol water distribution. The wetting angles of the investigated products in all cases lay between the values for cyclodextrin and niflamic acid, and the investigation of the system produced by the different preparation methods revealed that with increasing hydroxypropyl beta cyclodextrin content, The contact angles decreased, and usually a 1:2 and 1:3 ratios gave better results. Niflomic acid has a high partition coefficient value, reflecting its poor water solubility and high affinity for N-octanol. Its concentration in water at room temperature is around 26 microgram per milliliter, while that in N-octanol is around 50,000 microgram per milliliter. The N-octanol water partition coefficients reveal that the water solubility increase was generally better for the ternary than for the binary systems. The concentration of niflomic acid decreased in octanol and increased in water with increasing hydroxypropyl beta cyclodextrin concentration. The best partition coefficients results were 25 for 1 to 3 sonicated product, and we can conclude that the concentration of niflomic acid in water could be increased even 15-fold from 26 microgram to 422 microgram per milliliter. Also, we get very good results for the ternary systems. Niflomic acid has an acidic character, and its dissolution was therefore better in simulated intestinal media than in simulated gastric media. The dissolution of the cyclodextrin and cyclodextrin plus PVP containing products was in all cases better than that of the pure drug in both media. The dissolution behavior of the kneaded products was better than of other products in simulated gastric media. In this media, the dissolution increased in proportion to the cyclodextrin content in all of the kneaded products, where the 1 to 3 kneaded products gave the best dissolution results. The solubility increase was 4 and 5-fold as compared with the pure drug. Similar phenomena were observed for kneaded products together with PVP as a ternary systems. In simulated intestinal media, the use of PVP resulted in significantly higher solubility as compared with niflomic acid and the binary products. The saturation concentration was attained in 10-15 minutes. The maximum solubility increase around the 7-fold was observed for the 1:1, 1:2, and 1:3 ternary kneaded products. At the higher pH, ternary samples gave favorable results. Some of the kneaded products together, the PVP, dissolved totally, and in the first 10 minutes, a 20-fold solubility improvement was registered as compared with the pure niflomic acid. And finally, we investigated the permeability, the diffusion of the products measured by Sartorius apparatus, which was valuable only in the case of simulated gastric media. The diffusion was improved, the key D value around 1.5 and 2 numerical number could be reached, and the diffused drug amount to 5-fold. For all products, the diffusion was more advantageous than of niflomic acid in simulated gastric media. The next case study will introduce a special nasal powder formulations containing levodopa. Levodopa, as a gold standard in the treatment of Parkinson's disease, is usually administered per ounce, but its bioavailability is low. The intranasal administration is a potential alternative route in order to increase the bioavailability of the drug and treat the off period. Our aim was to develop levodopa-containing binary nasal powders with different excipients, such as cyclodextrin, by dry co-grinding process. The nasal route is an alternative administration possibility of drug transport to the central nervous system by passing the first-pass metabolism. The residence time of the drug on the nasal mucosa is an average of 20 minutes because of mucociliary clearance. The contact time or the residence time of a drug can be increased with mucoadhesive materials. Formulations of nasal powder result in high stability and higher dose can be administered also. The desired particle size range in the case of solid nasal formulation is between 5 and 40 micrometer because such particles are able to adhere in the upper airways with the best efficiency. Therefore, the drug particles should be grown to reach the given particle size range using different additives which can have grinding efficiency, the physical and chemical stability of the binary system, and the dissolution extent of levodopa. According to the literature, application of cyclodextrin could be good mucoadhesive excipients for nasal delivery. Moreover, cyclodextrin can open the tight junctions. For the preparation of levodopa excipients binary mixtures, a planetary ball mill was applied, where two process parameters were changed, the grinding time and the levodopa excipients ratio. The grinding was carried out for 60 minutes at 400 RPM as the different levo excipients mass ratios were co-ground. The effect on the average particle size, the dissolution, and physical chemical interactions was tested. The results presented that the optimized mass ratio and the process parameters was 70 to 30 in the case of levodopa and cyclodextrin, and it was found that the grinding process decreased the degree of crystallinity of levodopa in the ground products compared to the raw physical mixtures. These changes can be related to the nature of the excipients. The alpha-cyclodextrin and PVP had an intensive crystallinity degree reducing effect. It could be seen that micronization was achieved after 15 minutes grinding time. In vitro dissolution test was carried out in simulated nasal fluidum. These studies revealed alpha-cyclodextrin and polyvinyl pyrrolidine enhanced, but hydroxypropyl methylcellulose, PVA, and mannitol hindered the dissolution of levodopa compared to the raw materials. We developed the novel modified horizontal diffusion cell model following the nasal conditions in real time, in vitro, and ex vivo. So as a next step, we will investigate our nasal samples with this technique. Finally, of this study, we aim to measure the cytotoxicity of 6 pharmaceutical excipients, which could help to reach larger residence time, better permeability, increased solubility, and dissolution rate. The following excipients were investigated on human nasal septum tumor epithelial cells: mannitol, sodium hyaluronate, alpha and beta cyclodextrin, polyvinyl alcohol, and methylcellulose was used during the real-time impedance analysis and cytotoxicity test. No excipients showed toxicity at 0.3% weight per volume concentration or below, while 1% concentration, a significantly reduced metabolic activity was measured by MTT assay for methylcellulose and cyclodextrin. So if we apply this kind of excipients, we have used the lower concentration. And now let's see the last case study about the pulmonary application and using cyclodextrin. The most common treatment for respiratory infections involves oral or parenteral administration of high doses of single or combined antibiotics. Our goal was therefore to develop a carrier-free co-spray-dried DPI, dry powder inhalers product containing the broad-spectrum antibiotic, ciprofloxacin, and excipients such as cyclodextrin. Dry powder inhalation systems are formulated by micronized drug particles with aerodynamic particle sizes of less than 5 micrometer using particle engineering technologies such as spray drying using excipients. Cyclodextrin has potent and effective activity against a wide range of gram-positive bacteria and against mass gram-negative microorganisms, and it is often used in the treatment of inhalation, anthrax, and other lung infection. By the preparation of dry powder inhalers, a small amount of ethanol in aqueous solution was used. Then the fast evaporation during spray drying could affect the final size. Therefore, the feed solution was prepared by dissolving 1 gram of cyclofloxacin and different excipients at different concentration in aqueous solution containing 50 milliliter of diluted ethanol solution. The following investigation protocol was used, where the physicochemical characterizations containing micrometric properties and structural analysis, in vitro lung depositions, and cytotoxicity were evaluated. We produced a 2-3 micrometer sized co-spray dried sample with a spherical morphology could be predict a high lung deposition. Of course, compared with the raw material, the specific surface area also improved. During the aerodynamic investigation, the fine particle fraction, the mass median aerodynamic diameter, the emitted dose could be determined. The marketed DPI products presented around 20–30% FPF value. With spatial formulation and using of additive, or results could predict much more higher deposition in the lung. After 6 months storage, the tendency was the same. With further in vivo investigation, can we test the real efficacy of the different compositions. From cytotoxicity view, microparticles containing cyclofloxacin without excipients and with leucine or combination of excipients didn't change the impedance of lung epithelial cell monolayers in the range of 1 and 300 micromole concentrations, indicating no cellular toxicity. Pictures illustrate the kinetics of lung epithelial cell reaction treated with cyclofloxacin at 1, 10, 30, 100, and 300 micromole alone, or its formulations prepared with leucine, cyclodextrin, polyvinyl alcohol for 48 hours. We conclude that the applied concentrations are not toxic in DPI formulations. Dear colleagues, as a conclusion, it could be seen that cyclodextrin have important role in drug formulation. During our case studies, I introduced different technological possibilities to apply different kind of drug materials together with cyclodextrin through the peros, the nasal, and intrapulmonar application. We conclude that during the pre-formulation study with different kind of techniques, we can use cyclodextrin, for example, together with nitromic acid in a supergeneric formulations. We developed an innovative nasal powder formulation of levodopa together with cyclodextrin for treatment of Parkinson's disease. And then we use cyclodextrin together with an antibiotics, illustrated a novel possibility in treatment of respiratory tract infection, and we concluded that the presence of cyclodextrin, a stable, non-toxic formulation, could be reached with high lung deposition. And finally, it's time for the acknowledgement. I would like to thank your attention and, of course, the cooperation possibilities with Italian and Romanian partners. If you have some questions or suggestions, don't hesitate to contact me. Thank you again.